Methicillin-resistant Staphylococcus is a huge problem in both veterinary and human medicine. And when we're talking about methicillin-resistant staph, it's not just MRSA, but methicillin-resistant staph pseudintermedius and also coagulase-negative species. Term methicillin resistance is a little bit of a misnomer. We don't actually use methicillin anymore. So what we're really referring to is beta-lactam resistance. Whether it's an MRSA, an MRSP, some other coagulase positive or negative species, methicillin resistance is all about resistance to the beta-lactam class. So in order to describe the mechanism of methicillin resistance, I want to consider three scenarios. First are wild type staph, those which are fully susceptible to beta-lactam type drugs. If we treat these organisms with a penicillin type drug, so amoxicillin for instance, it's readily killed. This is a beta-lactam susceptible strain. Penicillin resistant staph, when exposed to the same drug, amoxicillin, produce a beta-lactamase called BLA-Z, which is able to degrade the drug and prevent it from having its activity. If, in addition to treating with our penicillin, we also include a beta-lactamase inhibitor, we're able to bind up that BLA-Z, allowing the amoxicillin to still destroy our bacteria. And so this organism is beta-lactam resistant, but beta-lactamase inhibitors restore susceptibility. In the case of methicillin-resistant staph, what we have are the production of altered binding sites within the cell wall. And these are simply not recognized by amoxicillin. It has nothing to bind to. Similarly, our beta-lactamase inhibitor has no enzyme to inhibit, and so these drugs remain ineffective against our methicillin-resistant staph. MRSA was first reported in 1961, so it goes back to the early-ish days of antimicrobial chemotherapy, first reported as silbenin-resistant staphylococci. Like I said already, methicillin resistance is really about resistance to all beta-lactams. There are a few modern exceptions, really drugs which are reserved for use in human medicine, things like ceftobiprole, but from the perspective of a veterinary practitioner, all beta-lactams will be ineffective. And the way that they have this resistance is through the MEC family of genes. The MEC genes code for an altered penicillin binding protein, PBP2A, which has a low affinity for beta-lactam drugs. And so our penicillins, our cephalosporins, and our carbapenems are unable to bind. And because they aren't able to bind, they're unable to inhibit peptidoglycan cross-linking and are simply ineffective. Because this mechanism of resistance does not involve the production of degradative enzymes, beta-lactamases, beta-lactamase inhibitors won't restore susceptibility. In addition to beta-lactam resistance, methicillin-resistant staph are frequently multi-drug resistant, and so we can lose multiple classes of therapeutic options. And when we think about what's most clinically relevant, we're really generally most concerned with our coagulase positive species, so MRSA or staph aureus, and MRSP or staph pseudintermedius. Again, I just want to reiterate that methicillin resistance is kind of a historical designation. We don't use methicillin anymore clinically, and we don't even test for methicillin resistance per se in the lab. We use other indicator drugs like oxacillin or cefoxitin. There's a growing family of resistance genes that confer methicillin resistance, so all within this MEC group. MEC-A is the classical gene, which likely originated from coagulase negatives, and the one that we've been dealing with since the 1960s. MEC-B is a homologous gene, which originated in Macrococcus caseolyticus. MEC-C was first identified in staph aureus, and subsequently was actually found sort of through a retrospective analysis of old isolates and was first identified in a staph aureus strain going back to 1975. And then finally, MEC-D is the newest member of this family, which also originated in Macrococcus species. On the right, I'm just showing an image of a Macrococcus caseolyticus, which I identified, displaying kind of an unusual phenotype. So we have a Kirby-Bauer disc diffusion test here. You can see there's growth right up to the oxacillin disc. We do have an inhibitory zone surrounding our amoxicillin clavulanic acid. And then interestingly, around the cefoxitin disc, we have this sort of target zone of inhibition where we have growth right up to the disc, but then further out, there's this inhibitory zone of clearing. It seems that expression of the MeCP gene requires a certain level of drug concentration in order to be turned on. So very close to the cefoxitin disc, we're expressing MeCP and we have resistance. In this zone here, where we have clearing further out from our cefoxitin disc, the gene is perhaps not induced. So it's not turned on and it's not expressed. And so we have susceptibility to the drug. Interestingly, both oxacillin and amoxicillin clavulanic acid seem to be more efficient at turning on that MeCP gene than is cefoxitin, resulting in confluent growth at a similar distance from the disc. So if we look at these two genera of bacteria, Macrococcus and Staphylococcus, on the left here, we just have a plate showing a strain of Macrococcus caseolyticus. You can see it's non-hemolytic. It has sort of a staph-like appearance. They're sort of creamy, whitish colonies. And then on the bottom, we have a staph aureus with its classical double zone of beta hemolysis. I've just drawn a sort of diagnostic tree to differentiate these organisms. So both Macrococcus and Staphylococcus are within our Staphylococcaceae family. Staphylococcus, of course, commonly differentiated based on the production of coagulase into coag positives and coag negatives. And then we can differentiate our most clinically relevant species, aureus, the intermedius group, and Staphylococcus. I mentioned before, MEC-A probably originated in coagulase negatives and was mobilized into Staph aureus back in the 1960s. MEC-C originated in Staph aureus, or at least the earliest reports of it. The oldest isolates that it's reported in are Staph aureus, while MEC-B and D come from sort of the other side of the family tree, the Macrococcus species. How do we detect methicillin resistance? Well, there's a few ways. Perhaps most simply is phenotypic resistance. So strains which are resistant to beta-lactam type drugs will raise our index of suspicion. For Staph aureus, we use cefoxitin or oxacillin resistance as an indicator of MEC-A-mediated methicillin resistance in our staph. For Staph pseudintermedius, we can only use oxacillin. Cefoxitin is not reliable for predicting the presence of this gene. We can also identify the MEC gene itself by PCR, and this can be done in any staphylococcal species. And then we also have a latex agglutination test where we're actually identifying the presence of that altered binding protein in the cell wall using latex beads coated with antibodies, resulting in agglutination reactions, which you can see in this lower image here. In both diagnostic and research settings, we can also use selective and differential media to get sort of a rapid presumptive identification of methicillin-resistant staphylococci. So oxacillin containing differential plates like this chromagar MRSA here can be a really good clue. Confirmatory testing, however, is required. So we still need to actually do either the phenotypic susceptibility test or identify the gene by PCR. So like I said, MRSA was first identified in people in 1961. That's Celbenin-resistant Staphylococcus by Jevons et al. It remained really a hospital-acquired pathogen for quite some years. These were really not very fit strains, so there was a large metabolic burden associated with carrying the MEC-A gene and its staphylococcal chromosomal cassette, and it wasn't able to survive outside of the highly selected healthcare environment. This all started to change in the 1990s. We saw the emergence of community-acquired MRSA or community-associated MRSA. So the fitness of these strains really increased. They adapted. They had compensatory mutations and actually novel chromosomal cassettes carrying the MEC-A gene, which imparted a lower fitness burden. It was less expensive for them to have this gene, which allowed them to survive in the non-selected community environment where there isn't necessarily a lot of antimicrobial pressure. In people, MRSA infections are associated with higher mortality rates and higher healthcare costs compared to the same infection caused by a methicillin-susceptible Staph aureus. In dogs, which I would say is the veterinary species where we're most concerned with methicillin-resistant staph, the same negative healthcare outcomes haven't been demonstrated. And whether this is due to the nature of the infections that staph oftentimes cause in dogs, frequently these are superficial. We see them as a cause of otitis and pyoderma, or there's just something different about the biology of staph pseudintermedius. We don't know. Here in our region, in Saskatoon, methicillin-resistant staph were first identified in animals in sort of the mid to late 2000s. So our first case of canine MRSA was recognized in 2006, and our first canine case of methicillin-resistant staph pseudintermedius was recognized in 2008. So I think it's important to just take a step back and recognize how common coagulase positive staph are. So 30% of you watching this video have staph aureus living in your nose right now. And that's entirely normal. It's entirely consistent with health. It's part of our normal microbial community. Similarly, in dogs, about 90% of them are colonized with staph pseudintermedius. So very, very, very common. Besides people and dogs, staph aureus has been found in many animal hosts. It's incredibly promiscuous and has a very broad host range. So we can find it all over the place in many animals which do not have any clinical disease. So it's normal. In 2008, I did a study looking at colonization of healthy dogs with coagulase positive staph. And we found that approximately 90% were colonized with staph pseudintermedius. This was looking at 3 body sites, the nares, the pharynx, and the rectum. About 10% of those same dogs were colonized with staph aureus. A single dog had MRSA, and at that point, no MRSP were identified, which, looking back on it, is quite interesting, and you'll see why in the coming slides. So historically, staph pseudintermedius in dogs here in Western Canada has been really, really wimpy. Resistance has been very rare. This is some data from a study that I did looking at diagnostic isolates of both staph pseudintermedius and staph aureus that were isolated from 1986 through 2000. We had a total of 60 staph pseudintermedius strains, none of which were methicillin resistant. And if we look at the frequency of resistance to other drugs, it was quite low. So only 7% resistant to penicillin, 8 to 13% resistance to our macrolide type drugs, around a third resistant to tetracycline, and 5% resistant to TMS. So pretty boring. Moving forward to 2008, so the same study looking at healthy colonized dogs, resistance a little more common, still infrequent compared to what we would expect to find today. So 40% penicillin resistance, low single digits macrolides, around 25% tetracycline resistance, and in this particular investigation, no resistance to trimethoprim sulfa. I should say that this population of healthy dogs was not treated with antibiotics. These were animals coming in for routine health checks. And so finding resistance in these is sort of an indicator of what's going on in the community in an unselected environment, particularly as compared to the table on the left, which are diagnostic isolates. So these are staph pseuds isolated from animals with infections. Our first MRSP we identified in 2009, and this is a really unusual case. So it was a 16-month-old neutered male pug dog with signs of a urinary tract infection, and multi-drug resistant Staphylococcus pseudintermedius was recovered, which was highly unusual, both because we didn't typically see a lot of resistance among our staph pseuds, and also that this was an otherwise healthy male dog where we don't typically see urinary tract infections. This dog had no identifiable risk factors, so it was otherwise healthy, had no previous antimicrobial use, and also had no contact with other animals who had been receiving antibiotics or people with identifiable risk factors. So this was a bit unusual, and we published this as a case report in the Canadian Veterinary Journal. When we looked at the susceptibility profile, it was really remarkable. So we had resistance to essentially all of the drugs commonly used in canine medicine, our macrolides, beta-lactams, aminoglycosides, fluoroquinolones, and sulfonamides. We had resistance to chloramphenicol, which is oftentimes used for treating methicillin-resistant staph in dogs. We had resistance to rifampin, which is quite rarely used in dogs for toxicity issues. If we go a bit further down, you can see some drugs which the isolate remained susceptible to. So nitrofurantoin, which anecdotally is not very effective against MRSP. And then, fortunately, this isolate remained susceptible to our last line of defense drugs, really important in human medicine. So vancomycin, daptomycin, and linezolid. So in this case, the only reasonable option for treatment was a tetracycline, and the dog was actually successfully treated with doxycycline and made a full recovery, which was great. This was another case that was recognized a couple of years later. This 6-year-old neutered male Great Dane who had a history of soft tissue sarcoma. He'd actually received radiation therapy at what became the site of the lesion here, and developed necrotizing fasciitis 3 years later. So this deep-seated, rapidly progressive infection along fascial planes between the muscles. MRSP was grown from the wound. It was aggressively debrided and aggressively managed, and the dog ended up making a full recovery. These early experiences really hit on the breadth of clinical presentations that we see with MRSP. More recently, a PhD student of mine updated the work done in 2008, so where we looked at colonized healthy dogs. And what this student found is that the frequency of resistance was much, much, much more common. So we're up into the majority of isolates resistant to penicillin and ampicillin, similar rates of macrolide resistance and tetracycline resistance. We're starting to see more resistance to other classes of drugs, so TMS, chloramphenicol. Remember, these are healthy, untreated dogs. And then most importantly, most impactfully, is that 9 of 78 dogs were colonized with methicillin-resistant staph pseudintermedius. So it's becoming very, very common in the community in animals who don't have risk factors for resistance.